Welcome to the MEF2C cast, a podcast dedicated to educating, supporting, and building community around MEF2C haploinsufficiency syndrome.
The MEF2C cast is for informational and entertainment purposes only. Views expressed are those of the host and guests and not necessarily of the podcast or its sponsors. This is not medical advice. Consult a healthcare professional for medical concerns.
Alrighty, hi everybody, and welcome to the MEF2C cast. Our guests today are Dr. Stephen Skinner, President and CEO of Greenwood Genetic Center in Greenwood, South Carolina, and Dr. Jessica Cooley-Coleman, who is a staff scientist at Greenwood Genetic Center. How are the two of you today?
Doing well. Thank you.
Doing great. Thank you. How
Good.
about yourself?
I'm good, thank you.
Good.
Can you start by telling us a little bit about how you became involved in sort of MEF2C research?
So I'll start and then let Jessica talk about herself. So I'm a clinical geneticist, been seeing patients for over 38 years now, and a pediatrician prior to being a geneticist. So much of my career has been seeing patients with rare genetic disorders, many of those patients, pediatric patients. And I do want to kind of make a disclaimer. I'm not really a F2C researcher as far as a primary area that I focus on exclusively, but MEF2C is one of those rare genetic pediatric disorders and adult disorders now that we see from time to time. And my actual first exposure to patients with MEF2C variants was actually by accident. I guess I had patients referred to me for a different condition that I was working with that sometimes has overlap, Rett syndrome. And determined that those patients didn't have Rett syndrome, but had something. And doing genetic testing found that they had mutations or variants in the MEF2C gene. So that kind of introduced me to some of the first patients I saw. But then had an opportunity, Dr. Chris Cowens, who I think you've had on the podcast, and many families that have children with MEF2C condition know him. Anyway, he was giving a presentation on our campus, and some of his grad students were here, and I was just talking to them about their presentation and asked them if they'd ever seen patients with the condition they were working on, and surprisingly, they said no. That kind of got us connected and talking and made it possible for them to actually see some patients with F2C haploinsufficiency, and then that led to some further collaborations with Dr. Cowan's lab, and I think actually led to Jessica getting involved with F2C research as well. So that's kind of how I got started in it. Yeah.
Yeah, and for me, my background has been in a laboratory. So I have been here at the Genetic Center since 2010, working in the lab, and then I decided I would go back for my PhD. So I joined Clemson University's Healthcare Genetics PhD program, and the timing just so worked out kind of perfectly where when I was joining the program, I think it was around the same time that Dr. Skinner and Dr. Cowan had just gotten connected, and people were wondering more and more about clinical features of MEF2C, and so Dr. Skinner thought, well, we need to kind of look at this a little bit more. Maybe we can involve Clemson University since we have kind of collaborated with them and graduate students from there. And then since I had just became a graduate student at Clemson, I think it just kind of made sense that I would be a perfect candidate to work on this project to help characterize this disorder from a clinical standpoint. So I was brought in pretty early on in my graduate career to help work on MEF2C and learn more about it, the clinical features and what we see in patients.
Yeah, that's great. I think some of your papers are really some of the first on MCHS really clinically at all. So there's probably a lot of parents in the community who have read your papers. Sorry, I
And
lost. Oh,
there were a lot of parents that were very instrumental in Jessica being able to write those papers too, because
yeah.
that was online questionnaires that she did that parents had to participate in that. So I know she's very grateful for the families being involved and engaged in that.
Yeah, that's
Very
one.
much so, yeah, and I had actually went to Charleston at the Medical University of South Carolina for I think it was the South Carolina Autism and Neurodevelopmental Disorders meeting that they had, and I was that 2019, I believe? And so I got to meet Dr. Cowan, that was the first time I met him and some of his graduate students, and several family members who have, kids with MEF2C or MCHS, they also came down to attend the conference as well because a few of the talks were going to be centered around MEF2C. And so while I was there, we discussed, I had this idea with my team, Dr. Skinner and my professors at Clemson, that we would develop a survey to give to these parents to gather information about their kids. And so while I was there, I was able to meet how many, 3 or 4 different parents, and they gave me their contact information. So they were very instrumental in helping me even kind of finalize the survey and do what we call piloting the survey. So they were the very first ones to take the survey before I even launched it to the rest of parents out in the world. So it was a wonderful experience.
Yeah, our community is very involved. That's one of the good things about it. So I'll maybe pose this question to you, Jessica. Steve, you can chime in if you're more than welcome to if you want to, but Jess seems to be a little more specialized on the MEF2C end of things. So can you just sort of give us an overview of MEF MEF2C as far as the gene itself and then the role the protein plays in the body?
Yeah. So, MEF2C is, one of our many, we have over 20,000 genes, and MEF2C, of course, is one of them. And normally, of course, we would have 2 copies of each gene, one copy inherited from mom, one copy inherited from dad. And with MCHS, so MEF2C haploinsufficiency, syndrome, the reason that it's called haploinsufficiency is if you have one copy that is either missing because of a deletion, or if you have a mutation or a variant on one copy, then it kind of renders that one copy non-functional. And so instead of 2 copies, you're left with one copy, and that's not quite enough to do everything that's MEF2C needs to do in the body. So the MEF2C protein is a transcription factor. And so transcription factors bind to your DNA and kind of help decide when to turn on and off other genes. So MEF2C is kind of playing a role to control, numerous other genes downstream. And so, if you're lacking some MEF2C, then certain other genes in different cells may not get turned on properly as they should, which might then lead to some of these features that we see coming up in a person phenotypically. Dr. Skinner, I don't know if you have further to kind of expand on that, anything I may have missed.
No, I think great explanation. I think the MF2C gene, as Jessica alluded to, regulates other genes, hundreds of other genes, and many of those other genes are involved in very early developmental processes in utero, in the womb, before the baby is ever born, but also continues to be involved in developmental processes after birth. And many of those are involved with the brain. And so that's why we see a lot of the neurological impacts from F2C haploinsufficiency, but also with the heart and muscles and with neural crest cells, which form a lot of the not only the brain, but also the face and some other structures. So I think Jessica did a good job kind of explaining that. In some of the earlier patients, that were first described actually had deletions where it wasn't just the MEF2C gene that was deleted or missing, but other genes. And at the beginning, it wasn't known what was the critical gene in that missing region of genetic material. MEF2C was known to be in that group of genes, but as more patients were identified and eventually a patient was seen that had a deletion that only included the F2C gene, it kind of put the spotlight on that gene as
Mm.
probably the causative gene for what we saw as MEF2C haploinsufficiency. And then later, there were patients identified with just variants just in that gene that impacted the function of the genes.
Yeah, that makes sense. Yeah, the analogy I've made about transcription factors before is like if you can imagine the MEF2C protein would be the blueprints for a house, and the MEF2C gene would be the machinery that makes those blueprints. So if the machinery that makes the blueprints is messed up, then in that everything downstream of those blueprints, everything you build based on those is going to be messed up too because the blueprints were messed up. So they're kind of cascading downstream effects.
Right,
Mhm.
for
But
sure.
yeah, we actually we just started doing t-shirts for the podcast, and one of them says ask me about my favorite transcription factor.
Oh, I love that.
So when does actually another parent asked me this last night and I didn't have a good answer and I said, "Well, I'll be talking to two geneticists tomorrow so I can get one." So when in de novo mutations, when does that mutation actually occur?
So, for the most part, it occurs before conception. So, it occurs when the germline cells are dividing, so either the sperm or the eggs. So, before conception. So in males, the sperm are constantly dividing on a daily basis. So that typically a change in a testes or in the sperm may occur, when those cells are actually actively dividing. Whereas eggs could even theoretically could happen even before the mother is born,
Mhm.
because those eggs are actually there before birth. But it's prior to conception for the most part. Now, we rarely will see variants in the genetic material that can happen after conception, in which case we sometimes get more of a mosaic pattern where there's a mixture of some cells that have the deletions, some don't, but that's very rare. So most of them happen before conception. Jessica, anything else you'd say on that?
Yeah, no, definitely. And really with when it comes to MEF2C, I think we've seen maybe one patient who had two different mosaic variants affecting the same amino acid of their protein, but with their two variants, it changed one to a different amino acid, the second variant to another amino acid. So together with those two mosaic, it made it, I guess, like math-wise, that they were about 50-50, like 50 normal and then 25% this variant, 25% this other variant. And so that was very interesting, but very, I think, rare, not something that I've seen besides that one other time.
Mhm.
So that would make me think in their case, it could have happened after conception, and then the cell tried to maybe correct to get back to normal in some way, but then miscorrected and formed a different amino acid than anticipated. So they had two different mutations at the same spot. So completely, you know, I think a rare situation, but mostly you have, just the one copy affected either by deletion or a single variant, and I think that would be prior to conception.
Gotcha.
And I guess kind of the genesis of that question a lot of times from parents, at least clinically, is parents wanted, is it something that I did during the
Yes.
pregnancy that caused this?
Yes, mothers especially, yeah.
Yeah. And this happens before conception, so it's not something that occurred during the pregnancy. And in particular, egg, it may even occur before the mom was even born. So nothing that was in mom's control. And unfortunately, these errors as cells are dividing and creating new cells, sometimes these errors happen in the replication process, and it's not that we can determine due to anything parents did or didn't do. It's just one
Right, yeah.
that happens.
Yeah, I mean, there's what, I billions of individual nucleotides in the human genome, something like that.
Oh yes, there's
There's
over 3
Yeah,
billion.
three
Yeah.
there's
billion.
an unbelievably massive number of opportunities for something to go wrong, and things just happen, and rare genetic disorders are rare by themselves, but when you aggregate all of them together, they're not so rare, you know what I mean? So, a lot of people feel, yes, this is you're one of 500 people with this, but they're it's just something that happens as part of, the sort of our reproductive process.
Right. Right, you're right. There's over 7,000 rare diseases that we know of now, probably more than that. And so as cumulatively, they are affect approximately 8 to 10% of the population. So
Right.
cumulatively, they're not rare, but
Mhm.
individually, they're very rare.
Yeah.
Yes.
So, I'm especially interested in your view on this, Steve, because I know you've done work with Rett and Angelman's and a variety of other things. How do you view the relationship between MCHS and autism? Is it syndromic autism, or how do you sort of conceptualize that?
Okay. Yeah. I'll try to answer that. I guess, autism is a spectrum to start with, I guess, in the current definition, and it's defined by really behaviors that are seen. So you have three broad categories. One is social interactions, just the back and forth when you're communicating or interacting, playing with an individual. How do you read body language? Do you read verbal and nonverbal cues? And how do you interact? So any so you have to have impairments in those kind of social interactions, but you also have impairment in language, and so just impairments in how spoken language is used, but then repetitive behaviors. If you think of MEF2C haploinsufficiency syndrome, a lot of patients have problems with language development. A lot of them have repetitive behaviors, and that impacts social interaction. So it very easily could get a diagnosis of autism if you have MEF2C. And many genes have been associated with it, over 400, probably more than that, but at least 400 that I know of. Uh, and each of those can have overlapping behavioral and developmental features that get you a diagnosis of autism, but they're not the single cause of autism. So I think MEF2C is one of those genes that can lead to what we call autism as far as the behavioral characteristics. And so, from a family standpoint, if you have a diagnosis of autism and you have MEF2C haploinsufficiency, I would tell parents certainly use that autism diagnosis to help you get services. Use that diagnosis to look at appropriate therapies for autism, ABA, and other things that might help. But also, you have to look at the MEF2 literature and research, and that approach for an individual with MEF2C and autism may be different from a patient with Angelman and autism or Rett syndrome and autism or something like that. So I think it is a it is one of the syndromic forms of autism from that standpoint. Uh, yeah, I don't know if that answers your question or not, but
Yeah, I mean, it's a complicated, it's I have some background in, like, taxonomy, and it's the same thing, you know what I mean? These are all artificial categories that we're making and then fitting things in, but some people view it very differently, and there's also the complicating factor of, in the United States, sort of, I don't know if political is quite the right word, but, like, that autism diagnosis is very important for accessing resources.
right, very, very much so.
So there's that part of it too, where it's sort of just a very practical thing that you need. And you talked about the genes that are associated with autism that are risk factors for autism, but don't necessarily cause it. As I understand it from things I've heard from Dr. Cowan, MEF2C regulates a lot of those
Right,
genes too.
correct. Yeah,
At the very least, I think it would be fair to say that having a MEF2C variation could increase your risk of
definitely.
having autism.
Right. Yeah, and in some thinking, and I wonder if with some patients with autism, it's a combination of genetic variants in multiple genes, and
Mhm.
maybe there's a threshold. Once you exceed a certain threshold of variants in certain genes, you present with autism. And your point about MEF2C, since it's regulating hundreds of genes, and many of those genes are involved in brain development and brain function, connections, communication. Now you've got one change in one gene, but it's affecting hundreds of other genes that are all in pathways that may lead to autism. So you can certainly see how that leads to going over a threshold into now having a clinical diagnosis.
Right, for sure. So what I wanna do is kind of not necessarily just this most as far as I know, the most recent paper you guys have put out on MCHS, I think it was a Gene Reviews from last year. But just kind of talk about natural history studies, why they're important, and then sort of the clinical description of MEF2C based on your findings. So can you start out, whoever wants to take this question, with telling us what natural history studies are and why they're important?
So, a natural history study one is basically looking at information that is collected with patients with a particular condition over a period of time. Ideally, it's done longitudinally, meaning over a long period of time, ideally with the same patients at multiple time points during that time, to really start learning what is the natural progression or the natural history of a particular condition, in this case, MEF2C. Because so often what gets reported in the literature is a snapshot in time of an individual when they were first diagnosed, and you know what their characteristics look like then, but you wonder, well, what happens 5 years from now or 10 years from now? So I think the natural history studies are critical for just better understanding and answering questions that parents have. Well, what's the future look like? What's going to happen when they're 10, 15, 20, 40? And it's only through these natural history studies that we start learning that. But it's also important, I think, to have baselines for future clinical trials. So a lot of times in clinical trials, pharmaceutical companies want to have, kind of a control group and a treatment group. And a lot of times parents don't want to be in a clinical trial if they're not going to be given the medication at some point. So if you have your already developed clinical control group through the natural history, sometimes you can use that information to conduct a clinical trial based on this natural history of 200 patients. This is what we typically see in our treatment trial. When we intervene, it changed the trajectory or the development of certain by this amount. So I think it's helpful from that standpoint, but also sometimes helps identify things that we don't know about currently. So, and I think Jessica saw this just in the survey. Families would tell us, well, you didn't ask about this problem or this condition. Well, we didn't know that was part of the natural history or the condition. So they would say, well, you need to collect information about this. So that's helpful, but that also helps in treatment trials to say, oh, what are the key features, problems, what are most important to family members for parents. So and we're developing treatment, what should we target to try to treat to improve? So I think they're critically important. One, to get information to help answer questions for families, to develop future research, clinical trials, but also just learn more about a condition. Jessica, you did a natural history study. Any additional information?
Yeah, I think that's I mean, you've said it perfectly. So, we want to kind of collect data that as much as we can to help characterize this, the syndrome. So, symptoms or medical information, genetic, test results, and just to help us gain insights into that syndrome and how it might progress over time, just exactly how Dr. Skinner described. And I think ultimately for us to, I mean, not only learn more and more about the condition, but hopefully develop treatments and improve patient care.
Yeah, I mean, excuse me, with sort of, the scientific method that you learned in like the fifth grade or whatever, um, it starts with normally with like asking a question. But I think the step that people forget is that you have to have some observation, some information to ask that question about. And that's what natural history studies do in a lot of cases, like whether you're studying an animal or a star, whatever, a disease, whatever physical phenomena, you just have to gather a certain amount of information about a thing before you can begin to tackle the problem. Um, one question I did have specifically about that most recent paper is it said that there were only 142 individuals with pathological variants identified when, like, in the Facebook group, they make you send proof of your diagnosis before they let you in the Facebook group, and there are well over 400 confirmed cases in that group now. So Is that just because there's a lot of variance or unknowns of unknown significance, or why is there that disparity between those numbers?
I think, the variance of unknown significance could definitely be playing into that. I also think it's on us geneticists and clinical scientists that when we diagnose, what we say is a pathogenic variant in MEF2C, it's on us to publish that information. So then when I'm going and writing up this gene reviews, I'm looking at what has been published in scientific literature.
Mhm,
So either, a journal or there's a mutation database that we like to reference. It's called HGMD, Human Gene Mutation Database. So I actually checked that one this morning just to see how many MEF2C, I guess, mutations are currently listed in HGMD, and they list 194.
mm,
It's on scientists to make sure, even though we've diagnosed a patient and we've given them a genetic test result like report, we also have to put that information out there for other scientists to be able to see, in the literature, in these databases, because then if it's not there, you'll definitely see that kind of difference in the numbers that you mentioned.
yeah,
But early on when you're first learning a association between a gene and a condition, it's easier to get that information published. Once it gets out there and there's been a number of publication, it gets a little more difficult to just every time you make a diagnosis to get it published.
right,
And so a lot of when Jessica was pulling data for her papers, she was getting published cases to make sure had clinical information she could include in her papers
mhm,
versus some of the databases. We know there are more patients in the world
sure,
than what are published in the literature. So
yeah.
a lot of the discrepancy is
Yeah,
that.
that makes sense. Yeah, that wasn't meant as a criticism in any way at all. I just would like to know. And there are projects like I don't know if you guys are familiar with Simon Searchlight.
Yeah. Yes.
And there's another one that another charitable foundation that just recently came online for MEF2C called the MEF2C Family Foundation. And I think they're working with a similar group. I can't remember what it's called, but who are making efforts to do things like that. But we just I know the community as a whole wants to know what they can do to help. So if
Right.
you only have access to 100 and however many it was cases, but there's 400 in the world. We want to know how we can help get that data
But
to
I
you.
think the connection between support groups, family groups, and scientists, researchers, clinicians is very important relationship as far as fathering information, but also uh, advancing research and eventually treatments. Because with social media and internet now, you can reach out across the world and you can find these patients and families that we don't have access to. Just we see what's referred to us, and we can see what's in the literature. So I think we need to continue to learn how to use that information to provide even more information and knowledge to advance care and management treatment more.
Absolutely. All right, so we'll go through some of the different categories of the clinical description of MEF2C, and you guys can talk about both, what you published and then what you've just seen personally from experience. So we'll start with developmental delay and or intellectual disability. And I guess my biggest question here, and this may be difficult to parse, I don't know. Um, how much, if at all, can you say that the symptoms of this are due to intellectual disability versus motor ability and patient's ability to actually express their intelligence,
Yeah.
if that makes sense?
No, it makes sense, and it's a great question, and it's one that I think we struggle with a lot of times, particularly in someone that's nonverbal and may have motor apraxia or dyspraxia. Are our testing mechanisms reliable enough and valid enough to give an accurate reflection of intellect or IQ, anything like that? And I think we are seeing in other conditions that sometimes we underappreciated or under-recognized how much is actually occurring in someone's brain and what they're processing, and maybe they're processing but have trouble getting that information back out, or maybe our tests don't pick up on what they're actually able to understand and comprehend. So I think it's difficult, challenging to get an accurate assessment a
Mm,
lot of times of what someone's real IQ level is. And I'm not a neuropsychologist and
right,
that
of
do
course.
IQ testing, but I mean, even though tests are based on, you know, features that we can test and normalize in a typical verbal population. So I think that's a good question and one that we struggle with. Just because someone has certain developmental issues, is it really an intelligence problem, or is it really just processing and communicating problems?
And it
I
makes
think
me
we
think.
also, yeah, do what?
I was just gonna say, I've sat in on some patients in the clinic with you, Dr. Skinner, and I recall even like a parent saying, they felt like their child could understand
Uh,
more than they were nonverbal, then so of course they couldn't speak it, but she said, I can look and see the look in their eyes, and I do think that they're understanding more than even I realize. So yeah, definitely.
and trying to find other ways for individuals to communicate is where we sometimes learn, hey, they really are understanding a lot more than we realized.
Mm,
If we can find some other methods of communication, whether it's eye gaze device or
mm.
communication, augmented communication, or other types of things too. So,
Yeah, I mean, we definitely feel that way with Leo, and I know there are a lot of people that use AACs, and there's some people that have had success with eye gaze devices. My friend Andrew, we mention him every episode, so our audience is probably sick of hearing him by now, but about him by now. But he is an adult who spells to communicate, and he clinically presents relatively severe.
mhm.
He's nonverbal, but he is he's a completely normal guy once he was able to start spelling and communicate adequately. And I there was a news story the other day of it wasn't MCHS, I think it was just autism, but there was a boy in Boston who in like 18 months went from being nonverbal, learned to spell, and like 18 months later he got accepted into MIT. So like, there it's you don't think about how literally almost everything is a motor skill until you know someone who has really poor motor planning, and then you realize how 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 much effect that it has on you. But so can you just talk a little bit about sort of generally the developmental and intellectual symptoms that we would see?
And certainly Jessica's paper outlined that. But I mean, most patients are nonverbal or have very limited oral verbal skills, and so that limits, I guess, our ability to assess a lot of those issues as far as development. But even with the motor, many many of the patients have difficulty walking, abnormalities of the gait, and so obviously that's a major motor milestone that everyone tracks as far as development. And then issues with the hand use and being able to do fine motor skills. That's another classic in pediatrics, always looking, do they have a pincer grasp and are they transferring objects and how do they hold a pencil, that kind of stuff. Jessica, I know you got a lot of that data in your paper.
Right. And yeah, I was going to point out, just like you did, the fine motor skills, because even that, if they don't have those fine motor skills, then, can they hold utensils to feed themselves? So, that makes it, more difficult, for them and on families and, depending how severe the person is, whether they can do that or not. But, that is something that we see often is kind of that they may or may not be able to reach those certain milestones of those fine motor skills.
Yeah. So feeding and gastrointestinal issues, I know Leo has like heartburn and constipation issues, that's pretty typical. There's at least one individual that I know of that has abdominal migraines. Do you have any ideas on what the causes of these symptoms might be?
On a very superficial level, and
Mhm.
Jessica, you may have more. I think, I always hear that the GI tract is like our second brain as far as the number of nerve connections, and there certainly is a strong interaction between the brain, the nervous system, and our GI tract, and a lot of communication back and forth. And I think we see anytime there's issues with the development of the brain, the nervous system, neurologic, we also see issues with the GI tract. And so I think it's very common that we'll see problems along the whole GI tract. And you mentioned, the motor difficulties, and that certainly affects chewing and swallowing. But I think it also affects just the muscles of the GI tract all the way down. But then just the nervous innervation of the GI tract can also cause problems with reflux, air swallowing, abdominal distension, constipation, diarrhea. You get the whole gamut of issues there. So, and then I think there's just an interplay that gets into some of the other issues between the nervous system, the GI system, and then the endocrine system and the immune system, and then you get a whole plethora of clinical symptoms we see over time. Uh,
Right, and I was just, maybe going to add, we know MEF2C is very highly expressed, the terms that we kind of use, of course, in the brain, but also in muscle tissue. And with the GI tract being smooth muscle, MEF2C would be or should be expressed there, should be playing a role. And then, of course, if you've got half as much MEF2C as you should, perhaps that's leading to, the gut motility issues, that muscle's not functioning properly. That's, kind of what came to my mind,
Yeah, that was the next
potential.
question I was gonna
Yeah,
ask. You took the words out of my mouth. Uh, gotcha, yeah, that
yeah.
that makes complete sense. So moving on to the next category, we have dysmorphic features. Can you talk a little bit about what those features are?
So, yeah, some of the features that we've seen, and it's not really been, I guess, like a particular, I guess, like not every patient exhibits these features, so we can't just, see these features and think, oh, they look like they have a MEF2C variant. But
Right.
patients who have variants in MEF2C, we've seen kind of a broad forehead, maybe thick eyebrows, deep set eyes, large ears with prominent earlobes. Those have been some of the features. So, I mean, a lot of those, I guess, are really featured around the face. Maybe Dr. Skinner can, talk about like when we do exams on patients, those a lot of times we're looking at those facial features and making notes of what we see versus what we think is, I guess, typical. So those are some that we've seen.
yeah. Yeah, I think when we're looking at physical features, and usually things like the face, sometimes the hands and feet, um, we're looking, are there recurring consistent features or patterns that we see that can identify something as a genetic syndrome or a specific condition. Um, and there's some conditions that, if you've seen them repetitively, you start saying, "Gosh, every patient with this condition looks similar to other patients who've seen this condition." Uh, I think what Jessica was alluding to with MEF2C, it's not a strong syndromic feature that every patient looks the same that has an F2C haploinsufficiency. We also have to be careful when we're looking at what we call dysmorphic features, that there are some things that are unique to the family too. So we
Mhm.
have to differentiate family features because we do tend to look like our moms and dads or somebody in the family a lot of times. So what's a family feature? What is a feature that's consistent with the particular condition we're looking at? I guess a classic that most people know what Down syndrome is, and most people once they've seen several individuals with Down syndrome, recognize a similarity between other people with Down syndrome, even though they may and are unique and different in their facial
Mhm.
features. There's some characteristic pattern. I don't think there's a strong of a clinical presentation of people with MEF2C haploinsufficiency, but they're what Jessica describes is kind of what's seen more
I
characteristically.
I have heard from some people, I can't remember who, but, like, there's a mark, like, in kind of the whatever you'd call this, like, the cleft between their collarbones is also really common.
Yeah, yeah. It's the suprasternal notch, and there's sometimes
Yes.
a pit or a little like a hole there almost. And actually, we haven't seen that often. Some of the early articles mentioned that. Jessica, I think there were even I think in your survey you might not have had any or one or two.
Yeah, I don't yeah, it wasn't very common that I can
But
recall.
actually,
Yeah.
one of the first patients I saw presented here was a patient that had a little suprasternal pit, and the clinician was an astute clinician, said, I think that's related with MEF2C. But
Yeah.
in most of the
Well,
patients I've seen with MEF2C haploinsufficiency have not had that.
right. Well, in MEF2C's case, that would be where those dysmorphic features come in, right? Like, one of the parents we interviewed, she had adopted her daughter.
Mm,
I think her daughter was, like, 12 or something, and she had never had any, like, real she didn't have a good life and, like, hadn't received proper medical attention related to this, and so they're trying to figure out what's going on. They had her genetically tested, and it came back like a possible MEF2C I don't remember what the clinical terminology was, but,
mm,
like, maybe a MEF2C variant, but there's apparently some company out of Australia, I can't remember their name, who uses AI to scan images of people with rare disorders and identify them that way. So she sent her daughter's image to that company, and that combined with her genetic disorder or her genetic findings kind of pointed them in the direction of MEF2C. So it may not be alone be enough to be diagnostic, but if you're trying to solve a puzzle, it might be a piece that can help.
certainly. Yeah,
Mhm.
and we've certainly used the facial recognition software. We've worked with a company called Face to Gene. I don't think they're from Australia, so it may be a different company. But use facial recognition to try to identify patterns that would point you to a certain potential diagnosis.
Right.
And yeah, sometimes it's helpful. You take any information you can get if you're not sure what the clinical diagnosis is.
Absolutely. So the next thing I want to talk about is epilepsy. And this, Leo has never had any seizures that we know of. So we've been very fortunate in this category. But that is, because of that, we kind of forget that, like, for a lot of families, the seizures is kind of the main characterization of this disorder. Like, it's their number one priority in the care of their child. So can you talk a little bit about sort of the prevalence of seizures, maybe if different types are more common or less common, and what you think the causes may or may not be?
Yeah, so seizures or epilepsy, whichever term you want to use, are common. I think a little over 80%, Jessica, in your study had seizures reported.
Mhm.
And there's not a specific seizure type that's uh, associated with MEF2C. I think almost every seizure type has been seen that we know about. And it makes sense, as Jessica described earlier, the MEF2C gene, is regulating lots of other genes, but many of those are genes involved in synapses or connections in the brain, and many of those involved particularly with the excitatory and inhibitory cells of the brain. So some that increase activity of communication, some that decrease. And if MEF2C is deficient, you get a dysregulation of that balance of exciting and inhibitory cells. And if you get too much communicate or stimulation of the excitatory cells, you get seizures. So it's a very common, and seizures are one, they're very scary when they happen. So it gets parents' attention, and it's one of those things can happen at any age. So I mean, commonly you have kind of peak area time periods when you might see it. In childhood, maybe 3 to 5, but then another peak later in mid childhood, but then again around puberty, you might see it. So it's something that parents worry about because you never know when it's going to happen. You never know what type of seizure, how long, how severe, where's the child going to be when it happens. Fortunately, most seizures stop on their own, but if you happen to be climbing on the top of a jungle gym when you have a seizure, that might be more dangerous to you. Or if you were swimming when you had a seizure, that may be more dangerous to you than the actual seizure.
Right.
Uh, and unless you have medication, there's limited things parents can do for the first one other than just protect the child from, you know, injuring themselves from where they are and from, choking or
Mm.
aspirating from something.
Yeah, that makes, and I know, a lot of, well, several parents that we've talked to said that when their seizures aren't controlled by medication, or some people use have used dietary intervention,
Right.
that like it just they're so frequent that they just interrupt their child's ability to function day to day, and then they interrupt their sleep, and it's not even so much the seizures themselves, it's just the things that they get in the way of that start to accumulate.
Right. And one question we get a lot of time with any genetic condition is there a specific seizure medicine that works best for MEF2C? And the problem is they have all different seizure types, and any of the seizure medicines may or may not work. And so it sometimes becomes almost a trial and error. Try this one, it doesn't work. Okay, we'll try a different one, and then you start adding, and then you get in the concern. Seizures are not good for the brain, and you don't want your child seizing, particularly multiple times a day. But the medications also have side effects and interactions, and so it's sometimes a delicate balance of trying to get seizures controlled without doing harm from the medication. If they're happening multiple times a day, yes, it interferes with day-to-day function, and you don't feel good if you're having seizures all the time, and then you get irritable and you don't sleep.
Mhm.
That makes seizures worse. So,
Right, yeah. And people that was well, that'll segue us into my next question. A lot of people with MEF2C just don't sleep very well. Leo, we've got him on medication that's kind of taking care of that, but
mm.
sleep disruptions are really, really common characteristic of this. Are there any like candidate causes for what may be causing that? Is it the same excitatory inhibitory imbalance or something else?
I think so. That and the just our circadian rhythm, our normal rhythm of, it's daytime, we're supposed to wake up, it's nighttime, we're supposed to go to sleep, I think is dysregulated too. So I think it's a combination of brain development, neural development, and it interferes with development of the normal processes that kind of let us know when it's time to go to sleep and when it's time to wake up.
Mm,
But it is a big problem, and then if you're not getting adequate sleep, that just makes a lot of other problems
right.
worse because it
Yes.
interferes. It can make seizures more prevalent. It
Mhm.
can make you fussy during the day. It can affect your attention span, and then it just kind of
Right,
cycles.
yeah.
Yeah, I was
Yeah.
just thinking that sounds like a horrible cycle. Like if you're having seizures, you don't feel good, you may not sleep well, and then if you're not sleeping well, you may be more prone to have seizures. So,
Yeah, exactly. Yep. Yeah, it's tough for a lot of families. Like I said, a lot of people have been able to control it with drugs. There are some people that have used a ketogenic diet and say that's the only thing that have worked. But in most cases, it is able to be controlled to some degree.
Right.
So one thing I want to touch on real quick is I know based on what I've read to the ability that I'm able to read scientific literature that MEF2C is heavily involved in cardiac development in utero. Do you see any cardiac manifestations in actual patients?
We do, and I
so
think Jessica,
yeah.
you saw a variety. If you want to, yeah, go ahead.
Right, yeah. So with some of the literature that I've read, there have been some patients that have, I guess, congenital heart defects reported. So those have included ventricular septal defects or double outlet right ventricle, some adult onset dilated cardiomyopathy. So there's been some defects. And, when I wrote this gene reviews, Dr. Skinner and I, and part of the gene reviews is kind of giving recommendations for, I guess, management. And so we kind of, because we know MEF2C is expressed in cardiac muscle tissue, and we've seen some of these manifestations in the literature. But I mean, it seems like they've been reported kind of at a smaller number, but we still say or recommend a cardiac evaluation, and having like an EKG and an echocardiogram just to be sure, if to see, did the heart form properly, or like what if something, is going on under the radar that we don't know about unless we check. Um,
And I guess I would say what we've seen and what's been reported in the literature so far is congenital heart defects. So heart defects are there at birth. So
mhm.
during embryologic development, certain parts of the heart didn't close or develop properly.
Mm,
But what we haven't seen any reports of, at least I'm not aware of something that develops after birth.
mhm.
So, you know, there are certain genetic conditions like Marfan syndrome where there can be a progressive enlargement of the aorta that causes significant problems later on. We haven't seen something like that that develops or evolves after birth. So, we screen, sometime after birth, early childhood, make sure there's not a hole in the heart or a valve that's not working. But if there's not a problem, we don't have any evidence yet that something's going to happen down the road as far as some new problem or some conduction problem or something like that.
Yeah. Yeah, I know, because in based on what I understand, in a lot of the studies on mice, it doesn't the MEF2C knockout actually doesn't seem to affect the heart issues that much, which is just a great example of why mice are not humans and you can only put so much stock into mouse studies. Um, are there any reliable diagnostic features in imaging that you've came across in your research?
I think the brain imaging, the MRIs and
Mm,
CAT scans show non-specific findings. So the corpus callosum, the connecting fibers between the two sides of the brain, may be thin. There may be some small, slight enlargement of the ventricles, the fluid spaces in the brain. Might be the overall brain volume might be less. There may be other subtle features, but those are kind of non-specific. We might see those in a variety of neurological conditions, genetic conditions. So it's not like with some genetic conditions that there's a feature that you might see on imaging that you say, oh, yeah, every time you see this, you think about this genetic condition. It's just another piece of the puzzle that kind of says that's seen and may be consistent with MEF2C haploinsufficiency.
right. Yeah, one of the other categories that you pointed out in your paper was recurrent infection, and we don't have to spend much time on this, but just to reiterate, so we did an interview with Dr. O'Sullivan. His first name is escaping me right now, and he's doing a lot of
Tim,
great Tim,
yeah.
yes. Uh, he's doing a lot of great work on that. For our listeners, MEF2C does seem to have some effect on the immune system, so if you wanna learn more about that, you should go back and listen to that episode.
And I'll just mention
Go ahead.
that was a collaboration that we were involved with that they were looking for particular samples from patients with MEF2C. And so we were able to connect with families to say, hey, we need some samples for this researcher that we had not met before in Los
Right.
Angeles. So it's just, again, emphasizes the importance of the family groups and having those connections so we can help others research
Yeah,
that
yeah,
they
and
have.
I was able to help connect them with Simon Searchlight, his graduate student whose name again is escaping me right now, but so that's why it's important that families participate in things like that as well, because that could potentially be what is for her a huge amount of new data to work with and work on things like that. One more thing I wanted to point out from that paper based on my reading is that the oldest patient in the paper was 69 years old. A lot of people have concerns about life expectancy and things because there's just not that much known about it. And because of how recently the technology has arrived, most of the people that are diagnosed are relatively young. I don't have any data for certain, but I could pretty confidently say that most of them are under 10 years old, I would say. But so I just wanted to point out that, obviously it's just one person. You can't pull too much data, put too much stock in it, but that there is at least one person who's lived a relatively long life with MEF2C, and also it's particularly underdiagnosed in older people, so there's just not that much data available on it. So that's just
Right.
something I wanted to touch on real quickly because I
Yeah, I
know
think
that.
that's an important point because, as you pointed out, the
Mhm.
technology wasn't available to make that diagnosis when those individuals were younger.
Mhm.
And when you get to be in the adult age, the access to services and testing isn't always there. So many patients that are in their 40s, 50s, 60s, or beyond, people at that point say, well, what's it going to change? So they don't do testing. And so we don't know
Right.
how many older adults there are out there with these various genetic
Mhm.
conditions. And that's the importance of the longitudinal natural history studies so that, eventually we can answer that question.
Right, and a lot of individuals, as we said before, this presents very similarly to autism, so they were just diagnosed with autism based on clinical symptoms, and then, maybe they're 40 or 50 years old now and they live in a group home setting and maybe they don't have much family left, they don't have anybody to advocate for them, so that they just never get the testing that
Correct.
they need. It's just kind of unfortunately a situation that happens a lot. So, it's going to take some time for the testing technology to become more widespread and also just for the base of patients that have been diagnosed to get older before we have enough data on sort of adulthood in these things.
But it does bring up a important point, I guess. There's nothing that we know about with MEF2C haploinsufficiency that would tell us the patient should have a shortened life expectancy as long as we treat the seizures and the feeding
Right.
issues and things like that.
Mhm.
And so families have to expect that their child is probably going to grow to be an adult and probably get to a point at some point they have to think about what happens when I'm no longer able to take care of my
Yes.
child.
Yeah. I can assure you that's a thing that a lot of families think about quite a bit. Can you talk a little bit about genotype phenotype correlations? First of all, what that means in layman's term, and then are there any reliable correlations within MEF2C, maybe between different variations or what have you?
Jessica, you want to
Yeah.
take
Yeah,
that or?
so in layman's terms, a genotype phenotype correlation is when we look at a person's specific genetic variant or their genotype and how that kind of translates into the observable like characteristics in that person, which would be their phenotype. For MEF2C, I mean, I would say we haven't seen genotype phenotype correlations like too many in specific, other than I can recall there was maybe a couple of papers that said if there were variants in this particular region of the gene, it's called the MADS or the MEF2 domain. So domains in a gene are kind of that region within the gene that when it turns into a protein, that region of the protein is what's responsible to say binding to DNA or binding to other proteins. So that, specific region. So when there are mutations in those domains within the MEF2C gene, individuals with those mutations may be more severely affected than, if a person has a mutation that's, further, you know, towards the end of the gene perhaps, because we do know that these 2 domains are kind of towards the beginning of the gene. And so we've possibly seen that, and I think there was maybe another study or 2 where they saw if a deletion or patients who had a deletion, including maybe the first small portion of the gene, might have this particular phenotype, which would be these cutaneous vascular malformations. So, kind of not really seen in most patients, but think of kind of like almost like a birthmark type abnormality on the skin. So if you're thinking like vascular, if you're thinking like, little vessels and how it kind of displays on the skin. So it might look like a abnormal type of birthmark.
Sure.
But not very many patients. I think we noted 2 papers that gave that potential genotype phenotype correlation. But
Gotcha.
yeah, I think in general, we've got our survey data and data from the literature, and um, I think it's just out there for other, researchers to maybe make more and more of these genotype
Right.
phenotype correlations that maybe just haven't been um, quite, I guess, elucidated yet.
There's there's kind of an idea I've heard before that maybe people with deletions or nonsense variations might have more severe symptoms than someone with a missense variation. Is that n- is there any a correlation there? Is that not necessarily the case?
I think I've seen that in the literature as well, and intuitively it makes some sense.
Right.
If you're missing the entire gene, or
Mhm.
in many cases missing that gene plus additional genetic material, uh, that you're missing one half of your MEF2C protein function. Whereas if you have a missense where it disrupts the function, but maybe you have a little bit of function that's still working, that's just not 100%, that may be milder than. So that's one of the things we're looking at. I mean, are deletions of the whole gene worse than missense, or are just single changes in part of the gene? I think that's a question we get a lot of time from parents. They want to know, do I have a severe
Mhm.
or a mild variant? And is there a particular variant that predicts whether you're going to have seizures or heart disease or something else, or and whatnot? And I don't think we've seen strong correlations that allow us to predict those specific details like that at this point.
Gotcha. Yeah. A lot, you know, I'm glad you're sharing what you don't know with us, but it's also very important that a lot of people hear what you don't know too, because I think a lot of people can jump to conclusions based on what very extreme generalizations, which is sort of all we can make right now based on the data that is available. It's good to hear that sometimes it's better to hear that you don't know because it doesn't put limitations on what might be possible.
Right.
So,
And one thing I always try to stress with families, MEF2C obviously is a very important gene that
mhm,
has very important function in the body, but it is one of 20,000 genes.
right.
And so all those other genes have an impact on perhaps the severity or the presentation of an individual with a MEF2C deletion or variant. And some variations in the other genes may make the presentation more severe or less severe. So there's
Yes.
a lot we know about that gene interactions because we're looking at MEF2C, forgetting about all those others. And then we also don't know what the impact of interventions that we make now may have on the natural history. So how important is early intervention, a specific early intervention? Are there other stimulations or things that will change that
Yeah.
severity picture too?
Yeah, and then epigenetics on top of that, like, I don't really understand epigenetics at all other than it's very complicated, so each case is exceedingly specific, and you shouldn't put too much stock into generalizations in any case or, this maybe this kid has a variation on the same place or something, so this is what we can expect. That's something that I thought at first, and you have to focus on doing what you can with your kid and
Your
not
child, yes. Yeah.
not compare
Uh,
to anybody else because, as I said, everything's so specific to individuals.
we're all unique.
Right. Yes. All right. We'll try to wrap this up here, guys. I know we're running up on time. So, in closing, excuse me, what are the next sort of immediate steps to better understanding MCHS? And what can families do to help?
Yeah. Well, I think we've alluded some. I'm getting involved with natural history studies, getting involved with the research if that's available, clinical trials if they become available. That's all a process of learning. I know families want to know the cure today and not have to go through that long process. I do think it's exciting right now. We're seeing so much happening in the realm of gene therapy, gene editing, and gene regulation, and maybe not specifically in MEF2C right now, but I think what we're learning from other conditions is going to be applicable. So, I mean, I think it's appropriate to have some hope and anticipation that there will be specific treatments and clinical trials coming in the foreseeable future. So I think that's exciting. But at the same time, uh, kind of a level set of realization that sometimes things don't go as fast as we'd like them. And some of these natural history studies, which may seem less exciting and slower, are important. And sometimes some of these clinical trials that are just trying to treat some specific features or symptoms are maybe just as important to at least learn from as we're moving towards, hopefully more definitive treatments like gene therapy, gene editing, ASO therapies that regulate expression and things like that.
Yeah, something I would add to that too is, like, especially because when you consider MEF2C, there might be, uh, I forget the technical terminology, but after a certain age, there might be diminishing returns on treatments too because MEF2C is so involved in development. At least that's the way I understand it. But MEF2C is severely underdiagnosed right now. The numbers I've seen from reliable sources are anywhere between 1 in 30,000 to 1 in 3,000 people actually have some MEF2C variation. Like, that's the true rate of it. So if it's 1 in 30,000, that's 60,000 people in the world. If you're one of these 500, let's call it right now, your data is exceedingly important and has an exceedingly large effect on all of those other people going forward. So even if it doesn't help your kid, which I'm fairly confident it will, but it will still help an incredible amount of people down the road. So it's really important to sort of think of the altruistic aspect of this and being able to help other families like yours, even separate from your child, I think.
I agree
Yes,
wholeheartedly. Yeah,
absolutely.
Yeah. Alrighty. So go ahead and wrap this up here. So you guys work at the Greenwood Genetic Center. It's ggc.org, correct? That's where the website.
that is correct.
Correct.
Are there any organizations, foundations, or anything who are funding your work that you'd like to mention?
Well, I mean, we have our own foundation, the GGC
Okay.
Foundation, that helps us. We're a not-for-profit organization, and
Mhm.
and we're trying to make sure that patients and families, predominantly in South Carolina, but really beyond, have access, one, to genetic services, so they get the evaluations and testing they need, but then trying to use that diagnosis to how do we move to treatments. We've been fortunate to get some support for various specific foundations that have helped with some of our clinical and research efforts. So
Mhm.
we're always happy to collaborate and participate with other foundations, family groups to try to address access, testing, and then clinical trials and treatment. Yeah.
Awesome. Alrighty. Well, thank you guys so much for your time. As always, our listeners can find us on Facebook. We just launched a new website, mef2cast.com, or they can contact us by email at mef2cast@gmail.com. Thanks, everybody.
Thank
Thank
you
you
for
for
having us.
having us.